时候就是演讲者有两个
嗯，
屏幕，所以要看一下他选的，这个没问题的。我们看到的是展示的那个。
这是我的啊，对，我就用这个屏幕吧。
OK，好，
嗯，行，
那行。
我要退出吗？还是
你觉得你想要暂时退出，你可以退出这个 share，你也可以就把它留在这儿。
嗯，好，都可以。那我就先放在这儿吧。
OK，good，好。
嗯，我去一下拿回来啊。
嗯。
金师姐好，金师姐。
哎呀，丁姐好，你们来的比我还早。
丁
哎，
老师，我刚才把你 mute 了，你可以打开
哦，
呀。
出来，对对对，是啊，
大伯你好。
你好，你好，
哎呀，好久没见啊，师弟们。
丁姐，丁师姐这个比以前更漂亮了，感觉。
我我是用的美颜啊，大哥。
啊
我这个啥，这个都到晚上了，我刚把孩子哄睡，我听着这个
啊，
睡前故事，我都想睡了。平时我早就睡着了。
我们我们我们家两个正在外面闹，他妈在弄他，然后因为我
啊
们不是
放
飓风嘛，
假了呀。
昨天
啊！飓风不要啊！是吗？不上
对。
还挺
学是吗？我说
吓人的，
这个是
昨天我都准备写应
疏散了。
急预案了。这风特别大，我们家后面有一个小孩儿那个
嗯。
滑滑梯啊，加那个秋千那种东西嘛，然后就
吹
被这个
翻了是吧？
风整个给吹翻了，那个都
嗯。
哦天，房子没事儿啊？
房子没事儿，对，
那
2周前我们
姐过去
也有一个。
了是吧？嗯，
已经过去了，
是吧？
今天过境了，已经到那个 Georgia 那边去了。
嗯。
哦，上海前几天也有台风，不过还好，这没有感觉没有那么恐怖。
对，主要是我们哎 Florida 嘛，你知道这个
我知道，
每年经常都要来这么几次。
对啊，你应该习以为常呀。
也没有，就是很讨人厌的你吧，大家希望好像这个心理很奇怪的，一方面就是说每次一看到这个来了，就希望他真的来嘛，如果不来还有点失望，但真的来了，你又其实也挺担心的，是不是？因为就像昨天下午，我觉得我那个玻璃都快被他们吹破了。
我们2周前也有一个
我们
飓风，还
嗯，
好它没有从我们这个城市登陆，在纽奥良兹那边，离得有点远，但是风是挺大的。
就小朋友可能喜欢啊，因为一刮风，我们这边上次也不上学，
嗯。
对啊，
有台
他们
风的话
开心啊，
就会
就放
是啊，
羊了呗。
对啊。
我们这一周
所以说你今天还没
5天
上
就上了
学是吧？
2天学。
嗯，哦，
挺好玩的小朋友呀。
Morning Charlie，Morning。
嗨嗨
毛林夏令。
，Charlie，听不见，
你太了，太了。
又见了
哎，关
，OK，good。
东贤，你现在在哪里啊？
我刚回来，
南京。
南京大学。
哦，去跟甘师兄一起了。
好久没看到阮石像了。对。
嗯，
对，我们也都是很多年都没有见，我跟丁丁都有好多年没见过了。
Oh, really？
很多年了，
嗯嗯，
多少年？很多年，
可能还上次还是估计
不止
16年那时候。
那是挺久了。
16年是啊，就在 在 Washington DC 是吧？在 DC 建的是吧？
应该是。
嗯，也不
对，这个
是一定，
看明
肯定
年，你看看明年能不能来参加我们 CADA
对
的
对对对，
年会？
我争取，
如果签证
我争取，
办了的话，
嗯，对，我早点去办签证，我跟李瑜他们一起去办，回头
呀，
我就跟
去
着
年李老师是
他就行。
来了，哎呀，
对，他来，他特地跑到东北去做的签证。
啊，沈阳那边
嗯，
据说当
是，
时名
好
额会少
像是，
一点。
嗯，因为他前年好像在上海那个啥，等了好久
那这个
没
签
等上，
证还挺不可预期的。我，
非常不可预期。现在哎呀，就比较复杂。
哎，对，
现在上海好像排队时间特别
上海
长。
排队时间长，对，
我上
所
次
以
上次去弄排第4个，我就不想弄了。
对对，那得到沈阳，沈阳好一点。
嗯，
Hey, Chiro, I haven't seen you for a long
哎
time.
嗨
Remember last time I visited Shanghai, we met
嗯
over there in your institute.
嗯，
it was 2020 something, right?
2017 or
17.
yeah, right, 7 years
It
ago.
has been very long time.
Yeah, right, right, right.
Time flies.
Yeah, time's fly. Hi, Binyan.
阿宾林，
哎，
嘿，
冰凌，你好，
你好，
哎，冰
周荣你
冰，
好。
哎，你好，
我们好像从来没有就是
没有
officially
线下
见过，
见过。
是吧？都
对
是
对对对，
视频。
是是是，所以明天一定要那个啥，
对你
争
早点办那
取去
个签证，
Chicago。是，
对，然
嗯，
后就
对
对，现在好像挺麻烦的。上次我们开会好像有几个人就来不及了，就没有来。
对，是很麻烦，我还没有办过，还是要因公护照的这种去办。我得问问李瑜，他
对，李玉老师好像，不过他因为办过之后就比较容易了，就是因为他因为之前就来过嘛，就是我记得他16年、17年的时候就来 CAD 的 meeting，可能他那个签证也还是10年之内都有效的那种。
没有啊，他之前去过，他前年不就没有办上吗？
是吗？
他每
如果
年对，
他当时
好像
用的
每年都得
是因公
弄一次。
来的话呢，sorry，如果他用因公来了，可能就没有十年。那10年签证
啊，
B1、B2那个就是旅游探亲的那个才有10年。
嗨，小勇，
嗨，小勇，
哎，你好，你
嗨，小
好
勇，morning。
哎，谢谢你
，Good
好。
谢谢你们
morning。
抽时间来参加这个 discussion，估计很多人现在都很忙啊，这又到了这一轮那个，
叫 Grant。
Yeah，
现在是美国 Grant 那个是
嗯，
吧？
十月五
下个
号，
Yeah,
星期
就是吧？哦，
六。
10月5
October
号，
deadline.
那很快了。
我已经放弃了这一轮，算了。
this is more important. Be the host.
要么我们再等2分钟吧，可能还有一些人会加入，然后你就可以开始了。
行，
大家好，打个招呼，好久没有上来冒泡了。
Morning，海滨，你好。
哎，海
啊，
边。
海边好，
还挺好啊。
你好，小杨。
海滨我们刚刚见过没多久。
哦，
对，
in 上海。
郑州 last year。
哦，OK。
稍微小一点，声音好，太大声了。嗯，然后 maybe we should get started.
Yeah.
Okay. So hi everyone, thank you for joining us for today's CAD Happy Hour seminar. So I'm the host, Xiangbo Ran, and today we are very happy to have our speaker, Dr. Qiurong Ding from Shanghai Institute of Nutrition and Health, Chinese Academy of Sciences. We also welcome our discussion panelists, including Dr. Charlie Dong and Zhihong Yang from Indiana University, and Dr. Wendong Huang from City of Hope, and Xiaoyong Yang from Yale, and Binglin Dong from Baylor College of Medicine. So I have been known Qiurong for quite a long time, back to 2006 when we were having PhD training in Dr. Yan Chen's lab. And starting from that time, Qiurong is always a role model for trainees from Dr. Chen's lab. Just one example, she published 4 first author papers in very different topics during her PhD. And I only published one. So I guess that's the reason why I'm always on the way to catch up with her. So after Qiurong finished her PhD in 2010, then she moved to Harvard University for postdoctoral training. At that time, she worked on the kind of hottest topics using TALEN and later CRISPR genome editing in stem cells to mimicking human disease. So again, she's very successful during postdoc training with 3 first author papers on high impact journals like Cell Stem Cell and Circulation Research. And in 2014, Qiurong moved back to Shanghai Institute of Nutrition and Health and started her laboratory there. Since then, she has won many awards and honors, including the Qinqian and Jieqin from China, and most recently, the CADA Young Investigator Award. So her lab's research has been published on high-impact papers like Nature Metabolism, Nature Communications, and Molecular Cell. So for our CADA members, you may be not very familiar with Dr. Ding's research. So today is a good opportunity to learn lots of cool stuff, including in vivo CRISPR screening in the liver, epigenetic reprogram, and zone-specific metabolic regulations in the liver. So welcome, Qiurong.
Thank you. Thank you for the invitation, for the very nice introduction. So before the talk, so I'd like to first thank the CADA family. So it is really my great honor to receive the CADA Young Investigator Award, so which is very encouraging to me. So thank you all for your support. So I did my postdoc research mostly on disease modeling with using stem cell models. So since independence, so we switched the direction a little bit. So my lab has been focusing on the study of liver metabolism. So today I will share you some stories about our discoveries about in the liver in identifying epigenetic regulators in liver metabolism. So we are interested in epigenetically how the liver cells respond to different nutritional, environment, or injury signals, and to maintain the metabolic homeostasis or liver function. And also we are interested in the diseased condition, how the liver cell lose their identity, and then whether we can target epigenetics to resolve the issue and to treat diseases. So taking the opportunity today, I will share you 2 stories. So one is a recently published one about histone phosphorylation. I'll just go briefly about the key points in this story. And the other is about a project we have been working on for a couple of years, which is still going on, about establish an in vivo perturb-seq platform in order to identify in a relatively high throughput regulators in liver metabolism and zonation. So the first story about histone phosphorylation, and in this study, we have identified histone phosphorylation as a key event in regulating liver gluconeogenesis. So you may all know that liver response shows very dynamic shows very dynamic changes in transcriptional activities in response to different nutrient or environmental stresses. For example, in the fast switch, you will see hundreds of genes that it changes in the mRNA expression levels. So we were wondering at that time, what are the alterations in the chromatin states during the fast switch? So we did sorry, I cannot see my title with this. Yeah. So we did a simple screening to this slide. So we did a simple screening with 4 antibodies. So against phosphorylation serine, phosphorylation tyrosine, and lysine methylation and lysine acetylation, so which are common post-translational modifications of proteins of histones or non-histone proteins. So we see, interestingly, we see a remarkable increase of the signal of serine phosphorylation in the liver tissue upon fasting. So we look at especially the one like around 15 kD. So where histone locates. So we look at the histone tails of histone 3. So that there are 2 serines in the histone tails in the phosphorylation S10 and S28. The phosphorylation of S10 and S28 has been previously reported that's mainly involved in DNA repair and also chromatin compaction during cell division or cell mitosis. So we first checked whether the phosphorylation of S10 and S28 changes upon fasting. So we're using the antibodies against these two phosphorylation sites. We see a remarkable increase, especially the S28. The phosphorylation of S28 remarkably increases upon fasting, and which reverses to the basal level after refeeding. And we know that during fasting and feeding, blood glucose changes. So we next investigated whether the blood glucose are the upstream signals that regulate the histone phosphorylation. So we fasted the animals overnight and injected the glucose. And this is a typical GTT experiment. And what we observed is that we see a significant negative corresponding of this histone phosphorylation signal with the blood glucose. So meaning that the histone phosphorylation changes according to the change of blood glucose. So we next wanted to see where histone phosphorylation locates in a genome-wide scale. So we did the CATAC and ChIP-seq analysis of histone phosphorylation. And we see that upon fasting, we see significant increase in the histone phosphorylation signals, and mostly around fast-induced genes, especially around those gluconeogenic genes. And you can appreciate here that the signal near the promoter of the gene. And then we want to contribute to this. So typical genes, the history that now.
Hi, Yanni. What?
Sorry.
So Choron lost her connection.
Sorry, for some reason I don't.
All right, good. No problem.
So where I was, which slide I was when.
I
So
think
I don't know.
you're
Yeah,
talking
this.
about the phosphorylation antibody responding to glucose.
Okay. So I almost finished my first story.
This will.
So this one, right?
Yeah, it's good. Mm.
Okay, I will start it from here. So basically, we did cut tag analysis to see where the histone phosphorylation located like in a genome-wide scale. So we see that the histone phosphorylation signal is significantly increased in the fast induced nearby the fast induced genes. For example, especially the gluconeogenic genes that you can appreciate here that those the signals are significantly increased in the during near the promoter or in the genes. And I think I will skip this story because it's already published. I will directly jump to the summary uh, for the time's sake because I'm more interested in my next story. So this is already published. So basically what we found is that upon fasting, the PK is activated, and then activated PK is recruited by crab. So crab will interact with PK and recruit the PK to specifically to gluconeogenic genes, and where PK will phosphorate histone 3 at the S28 site. And then the phosphorylated histone will be recognized by 1433 zeta, which is a reader for this histone phosphorylation. And this 14-33 zeta will recruit polymers 2 and to activate the transcription of gluconeogenic genes. And in the fat condition, we also identified a protein phosphatase 2A that can dephosphorylate S28 and then shut down the expression of gluconeogenesis. So I think our story that tells, because we know TF, the PK crab signaling pathway as the classic TF signaling pathways. So we think beyond that pathway, and upon fasting, histone phosphorylation will also, histone phosphorylation will enable a faster response to the signals and enable efficient transcription of these gluconeogenic genes. So this is also next I will talk about ongoing project. So about in vivo perturb-seq. So we have been developing this platform for quite some time, but still it's ongoing. It's still not ideal, but it's interesting. So I think I like to share you some of the results. So our lab have been putting a lot of effort in developing different high throughput genetic screens. So we think through the unbiased screening, we can get some clues and to identify key epigenetic factors and events in regulating liver homeostasis. And so the most common screening is in vitro screening. For example, we're interested in genes that are genetically associated with human lipids. So we customize a library and do some in vitro screenings with in vitro cell lines. And then you can identify some potential candidates that can regulate liver lipids. But they said for every in vitro screening, that you need to design a very nice readout. And sometimes you just cannot find a good readout for some specific biologic features you desire to do the screening. And the other thing is that because the in vitro system it's a very artificial systems, so for it's a lot of noises. So sometimes we just cannot. We will spend a lot of effort in validate those readouts in the in vivo system, and you just cannot avoid a lot of false positive or negative readouts. So we're also trying to do some in vivo screening. So a paper we published last year that we designed a very sophisticated in vivo screening to identify genes that involve in liver regeneration. So for this screening, we use liver regeneration. So it's a basically use cell proliferation as a readout. So for in vivo screening, I think it's you need to have some readout that can enrich for cells with the features you design. But for metabolic features, it is really hard in an in vitro setting that you can enrich for the cells. Because the cells, for example, the cells for tumors you can enrich for tumors that grows faster or proliferate faster or 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 metastasis or have enhanced metastasis. This is an enrichment for cells. But for metabolic features or for liver, for example, for lipid accumulation or something like that, just cannot enrich in an in vivo setting. So for this screening we use cell regeneration, but we cannot do the similar thing for the metabolic research. So we have been thinking about whether we can develop some screening strategy for metabolic features, for metabolic research in liver in vivo. So we look at the perturb-seq. Perturb-seq has been reported as early as 2016 and 2017. So basically, it is a combination of CRISPR screening and single cell sequencing. So for the perturb-seq in vivo, it's an in vitro system. So first reported in vitro. So for the screening that you infected the cells with CRISPR, so you get a pool of cells. So each of the cells will carry a sgRNA with one gene affected, perturbed. So you get a pool of cells with different genes perturbed in different cells. And then you subjected pool of cells to single cell sequencing. As for single sequencing, for each cell you can get expression profile of you can get the expression profile of mRNAs. So it's just like we did knockout experiment, we do RNA-seq, we know what has been changed after knocking out one gene. For this perturb-seq through single cell sequencing, you will get a lot of information about what the changes to the RNA profile after knocking out multiple genes simultaneously. You can get information of the changes to the mRNA expression profile after knocking out different individual genes in different cells. I'm still online, right?
Yeah, you're right.
Yeah. Yes.
Okay. So we are trying to put this system in vivo in liver study. So we designed to do this. We designed a AAV vector. So in this AAV vector, we have two transcripts. So this one is a known one. So it's a transcript. It's transcribed by the polymerase 2 In this non-transcript, we have a TBG-induced pre-expression to activate the Cas9 expression in liver, specifically in liver cells. And we have a venous expression, which will allow us sorting of it's a fluorescence protein, which will allow us to sort the cells with virus infection. And we also have a sgRNA cassette and a poly-A in the non-transcript. And this will allow us to catch the sgRNA information by the single-cell sequencing platform. And then we have our second short transcript, which is initiated by the polymerase 3. Then this will transcribe to the sgRNA, and then will formulate with Cas9, the Cas9 complex, which will target the gene expression. And with this AAV vector, the pep9 is that we have the AAV virus infected to the liver, and we will modulate the titer of the virus, and to ensure that the cells in the liver, so one cell at least, at most, have one virus. So the majority of the cells will not have virus, or just some of the cells were only infected by one virus, to ensure that only one sgRNA is expressed in one cell. And then we do the sorting. Use the fluorescence protein, and then we subjected the cells to single-cell sequencing. And then during the single-cell sequencing process, every cell is barcoded, and then we get the barcoded mRNA, and we then amplify the sgRNA information, sgRNA part, and we do amplicon-seq. And then from the amplicon-seq, so we can know the sgRNA's information as well as the barcode information. So based on this information, then we can project the sgRNA information to the single-cell sequencing results. So we will be able to know that for each cell, we will be able to know the sgRNA information, so we'll know that which gene is perturbed in this specific cell. And from the single-cell sequencing, we'll be able to get the changes of the mRNA expression profile. So basically, from perturb-seq, the input is the gene perturbation. So this is the same for every screening, in vitro, in vivo screening is the same. But the difference is the readouts. So for the perturb-seq, you don't need to design. You don't need to design the specific readout, and so you can just do this sequencing to get, so you can get more than the expression changes of more than 1,000 of genes. So this profile the changes in the profile of the expression of the 1,000 genes will give you a lot of information about the function of the cells, about the identity of the cells. So basically, this is the advantage of the perturb-seq. So to test whether this perturb-seq can work in vivo, so we started with a very small library targeting 11 HDAC family members. We also include some sgRNA controls. So we also subjected the animal to 4 different conditions: NCD's chill diet, high-fat diet for 3 weeks, 3 to 4 weeks, and fasting overnight, and the aging animal about 13 or 14 months. So this is so for each condition, we only for the sake of cost, we only did one single-cell sequencing. So for this is the number, this heat map is the number of perturbed genes per treatment and per gene. So for example, this 212 indicates that in the normal CHO condition, NCT condition, and through the single-cell sequencing, we have obtained 212 cells which carry the HDAC1 sgRNA. So those are numbers that in each condition with carrying other sgRNAs targeting HDAC2 or 3, something like that. So after we get this information, so we do analysis of the mRNA expression profile of these numbers of genes and then compared with other groups, and we can get a lot of information. For example, this is the COMBAS analysis about the changes of metabolic signaling pathways. So the red indicates the enhanced or activated metabolic signaling pathway, and the blue indicates the downregulated signaling pathway. And it seems that at different conditions, the changes are different during the changes are different. Oh, sorry, my sorry something. 我们家一个小宝宝现在醒了，我先把他哄一哄。I'm sorry, sorry.
It's okay.
So I'll get started here.
Yeah, we're good.
Maybe I was too loud to wake him up. Yes, so you can do a lot of analysis from this result. So here are just examples of 2 conditions. And basically that what we found is that under different conditions, actually you see different changes upon the perturbation of different genes. You can basically get a lot of information. You can compare for one specific gene, what are the or consequences of knocking out the same gene, or you can also compare on the same condition, what are the consequences of knocking out different genes. So of course, those are just results for about informatic analysis. So every result needs to be confirmed. So we first look at so we look at we did some we read some studies, and we found that some of the results we get from the perturb-seq can successfully validate some of the previous discoveries. For example, HDAC3 and HDAC5 previously discovered that in the fasting conditions, HDAC3 can regulate the liver gluconeogenic, and HDAC5 and regulates fatty acid oxidation. And what we can find from our perturb analysis is that after knocking out HDAC3, we see a downregulation of gluconeogenic genes, and also the decreased the lower activity of these gluconeogenic pathway activities. And also HDAC5, we can find that from the perturb-seq analysis that we can find if we in the cells with perturbed HDAC5 that the fatty acid oxidation expression of fatty acid oxidation is reduced. And so we're looking for some new findings. So we're mostly interested in HDAC6. So in the NCD condition, what we found that HDAC6 in the cells with HDAC6 perturbed, we see an enhanced regulation of triglyceride metabolic process. And then the specific analysis, the enrichment analysis, also told us that the triglyceride metabolic process is activated when HDAC6 is perturbed in these liver cells. And we also see a consistent upregulation of apolipoprotein genes that after HDAC6 knockout in the perturbed analysis. So in order to validate these results, so we did liver-specific HDAC6 knockout, and then we compared with wild-type animals. So those results are from the knockout animals. So we did HDAC6 specific knockout. So from these knockout animals, we see increased expression. We validated those results. For example, the increased expression of apolipoproteins, protein genes, although they are not striking changes, but we did see the consistent upregulation after knocking out HDAC6, which is consistent with the perturb-seq analysis. And we also see an increased blood triglyceride levels and decreased liver triglyceride levels. And we also find that knocking out HDAC6 will result in increased VLDL secretion. And this can explain why we get higher blood TG levels and whereas lower liver TG levels. We will still investigating the underlying mechanisms of how HDAC6 regulates this expression of apolipoprotein genes. So the thing is that you get hundreds from the perturb-seq analysis, you get hundreds of cells that with the perturbation of a single gene, and you get the profile of each cell. You get the RNA expression profile of each cell. So beyond the classic metabolic analysis, we think we can get more from these cells. So we tried to do we then did some analysis of liver zonation. We wanted to see whether perturb-seq can also be applied to look for regulators in liver donation. So liver donation is now well recognized. So because the liver have a very complex structure, and there is a gradient of oxygen and nutrient from the periportal vein to the central vein. So this gradient of oxygen and nutrient forms a liver zonation, forms the liver zonation, and in different zones of liver, and cells are metabolically different, and they are functional. They carry different metabolic activities. For example, in the periportal zone, the cells, the gluconeogenesis are higher. The activity of gluconeogenesis is higher in the cells in periportal zone, whereas the activity of lipogenesis are stronger. That is higher in the cells in the central vein, near central vein. So we did some analysis. So this is our first analysis. So basically, that for liver donation, that it's already known there are some landmark genes of liver donation. So for each zone, there are some landmark genes. So based on these landmark genes, based on the expression of these landmark genes, you can assign the cells to different zones, and then you can analyze. Then you analyze the gene expression changes changes between the cells in different zones. For example, this is what we get from the perturb-seq, the single cell sequencing results. That you can see that the gluconeogenesis gene is highly expressed in the cells in the periportal zones. And then the genes that encoding for enzymes in non-fatty acid synthesis is highly expressed in cells in the central vein zone. And then you can calculate zone scores based on the changes, based on the changes of these genes between different zones. You can calculate the liver zone scores, liver zonation score. So what we found that zonation is actually it's dynamic process. So that what we found that is that high-fat diet, 3 or 4 weeks of fasting overnight or aging can all cause changes to this zonation, to the zonation scores. And then for example, these are those I listed genes are PV genes, so they have a higher expression in peripodal zone. So you can appreciate here those are the expression profile in the NCD condition. And then this is the column close to NCD is a high-fat diet. And you can see a significant changes of these genes that in the high-fat diet condition, also in the fasting condition. Basically, there are sometimes some genes are the changes.
Okay, we might have some connection problem with Dr. Dean. Just wait.
Some histo staining of our liver slides, and the E-coherent staining are the peripodal vein marker. It's a green marker. So you can appreciate here that in the high-fat condition, especially we found some the peripodal vein, the zonation, the peripodal zone are mostly impaired in the high-fat diet condition. So based on this single cell sequencing results, you can also get an overall profile of the changes of these zone genes. For here that this is NCD. So here in the middle is peripodal zone genes, and you can appreciate here high fat diets cause impaired expression of those zone genes near peripodal vein. Also some changes near central vein, but the most differences happened in the peripodal vein. And then we can do analysis of these genes, enrichment analysis of these genes, that we found that in a high-fat diet condition, genes with impaired zonation are mostly about genes involved in drug metabolism, steroid biosynthesis, PBR signaling pathway. So based on the analysis or the calculation of zonation scores, then you can also calculate the changes of zonation scores after perturbation of each gene. For example, here, the HDAC family. So these are the numbers we calculated based on the changes of those zone genes. Now, for example, here is HDAC1 is 1.3. 1.3 meaning that the expression of these zone genes that among different zones have been enhanced. The difference have been enhanced after perturbation of HDAC1 in the NCD condition. And for these values lower than 1, it means that the expression of these zone genes has been impaired. So from this calculation, you can have an idea about the changes of the liver zonation by perturbation of each of the genes. So those are the bioinformatic calculations. So we wanted to do further validation of whether these whether these calculations are true. So to do this, to do validation, we picked up three HDAC genes, and for each gene, we generated the liver specific knockout animals. So for HDAC, so we specifically knocked out HDAC2, HDAC5, and HDAC9 in livers in animals, and we then did a single cell sequencing of these livers. So here are the cells we obtained from the single cell sequencing. And then so we also analyzed the information and also calculated the zonation scores. And it's very nice that we can validate the results we get from the perturb-seq analysis. So basically, the change of the zonation scores we obtained from the gene knockout animals are consistent with the scores we calculated from the perturb-seq. So meaning that the perturb-seq can really tell you some true information about whether a gene is a regulator of liver zonation. And here is some examples of the results. For example, HDAC2, that under the high-fat diet condition, perturbation of HDAC2 will cause impairment will impair the liver donation. So here what we found from the heat map. Here are the heat map from our knockout animals, that you can appreciate that the expression of the donation genes are impaired after HDAC2 knockout. And those genes with impaired donation after HDAC2 knockout are mostly enriched in linoleic acid metabolism, chemical carcinogenesis, and retinol metabolism. Of course, we still validate those genes with fish in liver sections now. And also you can specifically look at the genes you are interested. For example, those are genes with impaired donation expression in the white type animals, the NCD versus high-fat diet. And those are the genes that with impaired donation with after HDAC2 knockout. So you can specifically look at the genes, the donation pattern of the genes of your interest. And then we also tested the other gene is HDAC5. HDAC differently from HDAC2, perturbation of HDAC5 will cause enhancement will cause an increase in liver donation score. And what we find in the HDAC5 knockout, those are heat map of the cells with and the expression profile of the donation genes. And you can see that the enhanced expression of these donation genes after HDAC5 knockout. And those genes are involved in apnea signaling pathway, or progesterone-mediated oocyte maturation. I don't know what this means. But then it's interesting that we also found glucagon signaling pathway also have enhanced donation expression after HDAC5 knockout. So this is those are some preliminary analysis from the perturb-seq analysis of liver donation. So it's interesting. It's good that we can validate those results from the perturb-seq. So meaning that through the perturb-seq analysis, in addition to a lot of metabolic analysis you can do with the perturb-seq, you can also it also provides a potential powerful platform to analyze or to look for genes of to look for regulators of liver donation. So we also did some other tests with this perturb-seq with gene overexpression. So we wanted to see whether we can also use this perturb-seq analysis, perturb-seq platform to analyze different mutations and to analyze the functional consequences of different mutations. So we turn to HNF4 alpha. So HNF4 alpha is a key transcription factor in liver function. And in human genetic, there are over 100 mutations have been discovered in HNF4 alpha. And some of the mutations will cause the MODY1 disease, but the majority of the HNF4 alpha mutants, the function of majority of the HNF alpha mutants is unknown. So we wanted to see whether we can use this perturb-seq to analyze in a relatively high throughput of the function of these mutations. So in order to do that, we build up a plasmid pool with in total 150 plasmids, that including around 142 mutations that discovered in human genetics of HNF4 alpha. And for each plasmid, we have a barcode of the plasmid, and followed by the poly-A, so that we can catch up the barcode and also the mutation through the single cell sequencing. So just to briefly introduce the result. So for each HNF4 mutant, we will get an expression profile of the mRNAs. And then based on the expression of changes, a group of genes of the direct targeting genes of HNF4 alpha, we can calculate the functional score of these HNF4 alpha mutants. So based on the functional score, you will be able to know that whether this mutation is a loss of function mutation or is a neutral mutation. For example, we have some examples. For example, this is the score of the wild type HNF4 alpha. So those mutations with the dark blue have a very low functional score. So meaning that suggesting that those two mutations are loss of function mutation. And those mutations with green have a higher functional score. So meaning that those mutations could be neutral mutation. So to validate this result, so we generated the HNF4 alpha knockout liver, conditional knockout liver animals. And in these animals, we reconstitute wild type HNF4 alpha or different mutants. So because HNF4 alpha is very important transcription factors, knocking out HNF4 alpha in liver will cause a decrease of the body weight, and the animals with liver knockout of HNF4 alpha will gradually lose body weight and die. So what we found is that reconstitution of the wild type and the mutations of the green mutations, because those mutations are neutral mutations, they preserve the HNF function, the animals with these mutations will maintain the body weight and will not die. But those animals with reconstitution of the red mutations, which are presumably loss of function mutations from the perturb-seq analysis, they gradually lose the body weight. And we also did RNA analysis of the livers collected from these animals, and from the RNA-seq analysis, we also validated the results that those two mutations are loss of function mutations, and the RNA change profiles of these two mutations are similar to the knockout expression profiles of the knockout animals. So in summary, sorry, this is a very complicated story. I'm not sure whether I made myself clear, but in summary, that through our analysis, we think the in vitro perturb-seq is a useful platform that can be applied to screen for regulators in a relatively high throughput of liver function and zonation. And in fact, if the cost of single cell sequencing is is getting low because the cost of single cell is getting low, so you can do more single cell sequencing to cover more sgRNAs, so you can expand the sgRNA the numbers of sgRNAs in the library, so you can do screening with a larger with more genes. And from the perturb-seq analysis, we also get some clue that HDAC6 can regulate liver lipid secretion, and HDAC2 or HDAC5 are potential regulators of liver donation. And finally, I want to thank the members in our lab. So Zhao Yongshi who is now a independent principal investigator in Yantai 药物所. He did the phosphorylation story, and Dou who is a PhD student in our lab, who was a PhD student in our lab, but he's now he's doing postdoc research in Fudan. So thank you all for your listening. So I'm now taking questions. Thank you very much.
Yes, thank you, Sharon. Those are very exciting topics, and of course, it's a little bit hard to understand and to follow. So maybe we will start the question from our panelists, and we also have a few questions from the chat. So Charlie, you want to start with some comments?
Oh, sure. Great talk, Chiro. I think I really enjoyed the presentation. A lot of exciting data. So my first question is on your zonation studies using the HDAC perturbation analysis. So you mentioned a couple of them. HDAC2 is I think is a positive regulator, and HDAC5 is a negative regulator. So in terms of biological function of those two HDACs, I'm thinking about how they change the zonation based on their, the epigenetic function.
Thank you, Charlie, for your question. It's very important questions. So, so the reason we pick HDAC family is because they are important epigenetic regulators. So we think they will cause some, if you knock out those genes, will lead to some dramatic changes or some changes to the gene expression profile. So we don't know now for sure how HDAC2 or HDAC5 cause the changes of liver donation. The exact mechanism we don't know now, but we think it's through some regulation, of course, through some regulation of chromatin remodeling that we still need to look into deeply in the future.
I see. So I mean, so did you look at the overall cell functional changes, or this is some just some signature gene changes, because you use several marker genes to represent those specific zones?
Yes. So now we only have the changes of those future genes. We don't know exactly how HDAC2, the functional consequences of HDAC2, but from these changes of the signature genes, we are testing whether, because HDAC2 knockout has some changes to the steroid synthesis. So we are in the process of testing whether HDAC2 will affect bile acid synthesis or cholesterol synthesis. We're still testing this, testing whether it will have some functional consequences.
Sure.
We don't know yet.
Okay, that's great. My second question is on your HNF4 for screening. I didn't get the details. I'm wondering, did you check whether each mutant is only expressed in the single cells, or you may have multiple mutants in the same cells?
Yes, this is very important question that the key point of the paternalistic analysis that we don't want to get multiple one cell with multiple sgRNAs or multiple HNF for a mutants. So we did a serious test with the virus titers to ensure that each of the cell will only have one plasmid infected or one virus infected, so that only that one cell only have one sgRNA or one HNF for alpha mutation for mutants expression. So besides the virus, we type the titering of the virus, we can also do analysis from the single cell sequencing, and from the single cell sequencing analysis, you can also tell whether the cell has one or two or multiple expression of the mutations. You can exclude those cells with multiple mutations.
Okay,
you can do that by bioinformatics, but we try our best to avoid this situation.
I see. Great. Thank you very much. Great talk.
Thank you. Thank you.
Okay, then Wendong, you have questions?
Yes. Can you hear me, Chulong?
Yeah, Wendong. Hi.
Congratulations for receiving the CADA Young Investigator Award.
Thank you.
Also recently the JETCHI. Maybe you have more awards we don't know. Congratulations. Yeah, also great talk. And so you are using many state-of-the-art technology. So the second story, the perturb sequencing. This is interesting. I think this also indicates the beauty of the liver study. You know, you can manipulate the liver very easily using different technology. So is this technology commercially available, or you developed in your lab?
This is what we developed in the lab.
Oh.
So some perturb-seq plasmid is available.
Okay.
But you just need to engineer it to fit the study.
Okay, so your lab has done the engineering to make it fix to your projects. You're very
Yes.
impressive. Maybe someday I will ask your help to use that.
Sure.
Yeah.
Sure.
So the plasmid also is using the AAV, right, to Kelvin inject. Okay, so how efficient this to express this plasmid in the liver?
It's very efficient it's just very easy to target liver cells. Right. The AAV is very efficient to target liver cells, but for this study, we actually don't want that efficient. So we titer the virus to a relatively low titer. I think we use 10 to the power of 9 for each animal. So usually if we do liver specific gene knockout using AAVs, we usually use 10 to the power of 11. Wow. So we toned down the viral titers to 10 to the power of 9 to ensure that the cells don't get too many viral particles.
Okay. Yeah. So fantastic. Thank you Thank you very much. This is only my question. Congratulations.
Thank you. Thank
Yes,
you.
thank you.
Thank you, Wendell.
Now let's 小勇.
Yeah, Chorong, the wonderful work, and also congrats again for the two major award, and also we heard like you get with all those award, you also have a new one. I don't know how old is it? So congrats
Thank
for
you,
all the
Xiaoyong.
achievement you have. So anyway, uh the work for the first part, and you that's very nice, which you look at their majorly glucagon stimulate their phosphorylation of histone S28. So whether but you also show low glucose alter the histone phosphorylation on that side. Do you know the pathway that should not be the glucagon, right? It should be something else. That
Um,
that's the initial data. Remember to show the data. Maybe if I get it right, you actually there was a you saw the glucose, low glucose can change the phosphorylation, histone on
you're
that
right.
side. Yeah. So in that
Um,
sense, that should not be pKa crab dependent, or should you be? I don't know. So
in our study, we found it is pKa. That is the kinase that activate that phosphorylate histone. And
yeah,
then
but
we
even low
the
glucose, low
crab,
glucose itself can. You mean
yeah, low glucose.
low glucose itself can increase the pK. I mean that's in vitro,
Yes.
right? That's in vitro.
That's in vitro. Yeah. No, that's in vivo.
Oh, that's in vitro. Okay,
In vivo,
I
yeah,
didn't
in
get it
vivo.
right.
In
So I thought
vitro,
in
there's no,
vitro.
yeah,
Okay,
yeah.
all right.
That's
Sorry
in
about that.
vivo.
Okay, right. Uh, yeah. So for the this is a very powerful perturb screening. Just wondering whether with that one, did you look at how this siRNA sgRNA target other cell type in addition to hepatocyte, particularly for the low titer? Do you know look at the impact of other? uh, Do you have any sense about you know how the other non-permanent cell, how those cell got infected, and the functional consequence of this
Uh,
sort of sequencing
yeah.
can sort it out?
Yeah. So no, this perturb-seq, because with virus we have a TPG promoter, so they only can activate Cas9 in hepatocytes. And then
Oh, I see.
we sorted, yeah, we also did sorting. And now we also so sorting that we also did analysis, of course, from the single cell sequencing. We also did analysis of the cell types. So they are all hepatocytes.
I see.
There are
Yeah.
no non-parenchymal
No other cell. No.
cells.
Okay. All right.
Yeah.
Gotcha. All right. Thank you.
Thank you.
Thank you, 小勇. Then Binging, you have any comments?
Children, can you hear me?
Yeah. Yeah, Bini. Hi.
Actually, some of my question has been answered. So the last question I have is that when you feed the mice with high-fat diet, and then probably the pathological change is not that obvious. I'm wondering, like in MASH condition, when hepatocyte number decrease a lot, and then there's a lot of like immune cell like infiltration, and then can you comment if the donation could be changed? And then how do you use your methods, to apply on these condition? It's like fibrosis and all these like things going on.
Yeah, I think you raised very interesting question. We don't. I I think for sure the donation will change because the donation is really what we found is really a dynamic thing. Because fast overnight will cause a dramatic change to the donation. Like if you, of course, it's the donation. I mean, donation is the donation of the expression profiles of these donation genes. But for MASH, I think it's a fibrotic livers. It's probably not that easy to perform the perturb-seq because you need to deassociate the cells and sort it out the cells. I don't know, but we may have a try with that.
And usually the fibrosis, like starts from the central vein, right? And then it's like
yeah.
There is like a
the
progression of
steatosis.
different grade of fibrosis. So this also like might affect the zonation change. I mean, they just like my expectation, so I don't know.
Yeah, I don't know whether the fibrosis starts from the central vein, but the steatosis still starts from the central vein because central vein is a major zone for the lipogenesis. But I'm not sure. I think it would depend what type of the injury the liver cells have.
Yeah, I'm talking about the MASH.
The MASH, right?
Yeah,
Yeah,
MASH
MASH. I
fibrosis,
don't
not
know.
the
I think it's
injury induced or
yeah.
like bile duct fibrosis. So probably all different.
Yeah. Yeah.
Yep.
I don't know. I think it's interesting to look at.
Yeah,
Yep. So
more likely in MASH, all the zonation is greatly
Change.
impacted.
Yeah.
Yeah, it's, at least for those, because most of the markers like amino acid gene, they change a lot during NASH. And okay, then we have our last panelist from Zihong Yang from Indiana University.
Hi, Churong, and congratulations
Hi,
for
Hong
this wonderful talk. And it's very high quality research. And to continue the question from Bingling, and I'm also have a question regarding the liver zonation. And in your different model with the fasting and high-fat diet and the aging model, did you see any difference among this model on the zonation expression pattern, especially like which model mainly changing the genes in the central vein or the portal vein, or the expression pattern changing the central vein and the portal vein could be different in these different models?
Yes, yes, for sure. We see very different changes. Let me see. I just briefly go through. I think this slides. So we actually see very dramatic change after fasting overnight. So those are
Oh,
the
yeah,
so
here.
those are the scores of the liver donation scores at different conditions. So we have a pericentral score, periportal score, and the distance. These score, of course, those are like different. Those pericentral score, and these are the NCD. So what we found is that fasting is cause some major differences in the fasting and the aging are all cause some differences in the periportal zone. And those are I'm not sure.
Is that
Yeah.
means in the portal zone area and during the fasting and most of the gene were downregulated or they were
It's not
light regulated? That
yeah, it's
score
not I didn't explain very well. So it doesn't mean those genes are the lower score doesn't mean that those genes are downregulated.